Hello students, myself Dr. Madhusmita Borthakur, Assistant Professor, Department of Applied Biology, University of Science and Technology, Meghalaya. Today, we are going to discuss about an important topic of the developmental biology. This topic is for BSc Biotechnology students. The topic is the cytoplasmic mixing and the formation of a granule. Yes, I have said this is a basic of the developmental biology. In the developmental biology, we learn about how these embryos are formed from a single cell. From that embryo, how it develops into a tissue, how from the tissue it develops into an organ, and what are the roles of the other components it plays during its further development. So before that, we learn about the cytoplasmic mixing and the formation of the great reset, how this embryo is to form. Means this topic is important in order to determine which side of the embryo will develop into the face, which side of the embryo will develop into the tail. Tail means the dorsal side, face means the ventral side. Try to remember, face the ventral side, tail is the dorsal side. Means our back side is the dorsal side, our front side is the ventral side. Okay. Coming to this topic, cytoplasmic mixing. Before the cytoplasmic mixing, we have to know what is cytoplasm, right? So whenever we say it as a cell, what we define it as a cell? Normally, as per the cell biology, it's the structural hereditary unit. Fine, the basic unit of life, that is the cell. But in terms of the developmental biology, we say something which is cell membrane and a controlling element. The controlling element is none other than the nucleus. Okay. So out here, in here, we consider the cytoplasm of the egg cell. We consider here the cytoplasm of the egg cell. In this egg cell, egg is ultimately a cell having an outer layer with a nucleus. Fine. Apart from that, it also do have a semi-liquid fluid substance in it. Okay. And this egg, all the materials for the growth and development is present within this cytoplasm. All the materials required for its growth and development is present within the egg cytoplasm. It helps in synthesize or absorb protein. Always remember, protein synthesis takes place in the cell cytoplasm. Inside the nucleus, what happened? The DNA will be formed the mRNA through the process known as transcription. But in the cell cytoplasm, translation takes place. That is, the protein formation takes place. This also acts as a reservoir for the developing embryo. The cell cytoplasm often seen act as a reservoir for the developing embryo. As it provides protein, the early embryonic cell needs a supply of energy and the amino acid, which is accomplished by accumulating yolk protein during the meiotic cellular division of the oogenesis. Oogenesis means the oocyte, which will later develop into the egg cell. Okay. Inside our body, these gametic cells, it is the oocyte in case of the oocyte, which will later develop into the egg cell. Then the sperm cells, this will undergo the process of meiotic division. Meiotic division means the process of reductional cellular division. At that time, the early embryonic cell needs a supply of energy and the amino acid, which is accomplished by accumulating this yolk protein, which we consume. What ultimately it will help in providing the energy and the amino acid. Amino acids are the building monomers of the proteins. Okay, whenever we say, our parents used to say, have consumed egg. Egg is having the yolk is having the nutrient part, the energy part, and the amino acid will supply. The early embryonic cells need to supply energy and the amino acid, which is accomplished by accumulating the yolk protein during the meiotic cellular division in oogenesis. Then in the cell cytoplasm, also there are ribosomes, because without ribosomes, proper protein synthesis won't take place. So early embryo needs to make its own protein. Amphibian produces up to 10 to the power 12 ribosome during this meiotic cellular division. Apart from that, they will be needing a template, means the messenger RNA, where the ribosome will stick and it will form the protein. This mRNA will remain dormant until after formation. Then apart from that, the cell cytoplasm also has certain factors. We say these factors none other than the proteins. That is the morphogenetic factors. We have not named that proteins. We say it as the morphogenetic factors. That is the molecule that direct the cellular differentiation. So you can ask me, what is cellular differentiation? So in the developmental biology, cellular differentiation is a process which helps in differentiating the cell into a more specialized cell. So whenever I'm saying more specialized cell means whenever in case of the developmental biology and embryo forms. How does this embryo form? When these sperm cells will encounter or move inside the nucleus of this sperm cell, I'm saying the nucleus, not the cytoplasm. Remember, the nucleus of the sperm cell will move inside the egg cell at that time in the egg cytoplasm. Before fusion, it is known as pronucleus. Before fusion, it is known as pronucleus. Okay. After fusion, whenever the zygote is formed, means n plus n number of chromosomes from the sperm cell, n numbers of chromosomes from the egg cell. It will form the zygote. Now, zygote is a one cell, fine. Then from one cell, it will form multiple cells from 1 to 2, 2 to 4, 4 to 8, 8 to 16, 16 to 32. In 32 age stage, it is known as 8 to 32 stage, it is known as morula. Morula means it looks like a mulberry, the bunch of mulberry. That is why it is known as morula. Okay, now from that 32 to 64, this stage is known as the blastula. The blastula means cell has developed a specialization, cell has attained its fate, means which cell will be forming the ectoderm, which cell will be forming the endoderm, which cell will be forming the mesoderm that will form. Means ectoderm will be responsible for forming the outer layer. Always remember, ecto means outside. Okay, ectoderm is responsible for making the outer layer, that is the skin, hair, eyes, then nose, then nails, then mammary glands, then central and peripheral nervous system, then neuronal cells and all this. Then it will form the endoderm. That is the neck region, the region inside the neck. That is the trachea, the respiratory system. That is the oesophagus, the food pipe, the digestive system, the thyroid gland, the parathyroid gland, the tonsils, the thymus, the liver, pancreas, lining of the urinary bladder. All this will be formed by the endoderm, that is inside. The mesoderm, the middle part, it will form the vesicles, it will form the lymphatic vesicles, it will form the various muscle cells like the cardiac cells, like the skeletal muscle cells, like the smooth muscle cells, like the reproductive, like unit, like reproductive organs, like testes, like ovaries, the urinary system, like kidney, urethra. All this will be formed by the mesoderm. During this time, it is known as a specialized cell. And all this will attain from a single mother cell at that time we say it as a zygote. And the process of zygote development into a cell when it will attain a fate of a specialized cell, the process of zygote, development of a zygote to this ectoderm, endoderm, mesoderm. This whole thing is known as the differentiation until the cell is specialized in the blastula. Okay, after blastula, when it will form, develop into an organ tissue, at that time we say it as a gastrulation. Fine. So till that it is known as a differentiation, and differentiation does not take place abruptly. Inside the cell, various biochemical pathways are there. Through various biochemical pathways, cellular differentiation will be taken place. Apart from that, in the cell cytoplasm, certain protective chemicals, DNA repair enzymes are there, which will help to repair the damaged DNA, which will help to repair the damaged DNA. So we'll remember in the cell cytoplasm, ribosomes, tRNA is there, various proteins are there, various messengers, RNA are there, then morphogenetic factors are there, along with that certain protective chemicals are there, which will help to maintain the cell, the proper functioning of the DNA in this cell cytoplasm, it's egg cytoplasm. Now, in this, in the early development, a zygote is been divided into two hemispheres. Fine. The animal pole and the vegetal pole. Means egg cells we are dividing it into, now I'm so sorry, it's like the egg cell we are dividing it into two hemispheres. One is the animal pole, another is the vegetal pole. Into the vegetal pole, the yolk concentration is very high. See, can you see it's the vegetal pole is something whitish in color because the yolk concentration is very high. Since there is a very high yolk concentration, it's very light components are there. Very light, minimal components are there. Okay. Whereas in case of the animal pole, the yolk concentration is less. Since less amount of yolk concentration is there, it is a very darker component. Now, the two hemisphere is important in order to maintain the cellular polarity of the egg cell. Whenever I'm saying the cellular polarity, we have to remember one point, that polarity means which side will be the face, which side will be the tail, which side will be your ventral side, which side will be your dorsal side. This is something, the upper part, the dorsal side, this is the face side, having the teeth, this is the ventral side. Okay, the two hemispheres important in order to maintain the cell polarity. Now, the ectosome of the sperm cell consists of a lytic enzyme. or you can say it is a digestive enzyme. What does it enzyme is? Hyaluronidase and acrosine. Means the acrosome, the head part of this sperm cell, the head part of the sperm cell consists of a lytic enzyme, that is the hyaluronidase, and acrosine, try to remember that, which break down the outer layer of the egg cell, that is the zona of pellucida, forming a deeper depression, forming some kind of a depression, that is, or you can say it's a pore, it's a germ pore, forming a germ pore for entry of the haploid sperm nucleus into the egg cytoplasm. At that time, fusion has not taken place. So whenever the sperm's nucleus will enter inside the egg cytoplasm, the egg also have its nucleus. The sperm, whenever it will enter at that stage, we say it as a pronuclei. Fusion has not taken place. Means it's still N and N. N from the sperm cell and from the egg. Okay. And the zone of entering of the sperm nucleus, the animal pole, is considered to be the ventral side. Remember, animal pole means the mouth part. Just diagonally 180 degree to this is the dorsal side, or is the posterior part, the back, the nervous system. Fine. So towards the mouth side is the animal side, like towards the point, like where germ pore is there, that will be the animal pole. That will be having the face, that will be having the ventral side. Just 180 diagonally to it is the dorsal or the posterior side, that is the back or the nervous system. Now, the outer boundary of the egg cell consists of a loosely bounded cytoplasm, which leads to its movement through the motor protein and the cytoskeleton inside the inner core, forming the cortical region, which are tightly bound. Okay. Whenever the sperm cell will enter its nucleus, at that time, there need to be some kind of mixing should be takes place between the mixing of what? Mixing of the cytoplasm. From the animal pole to the vegetal pole. Again, from the vegetal pole to the animal pole. There need to be some formation of some or mixing of the nucleus, mixing of the cytoplasm is necessary for the formation of a proper zygote. Okay, the outer layer of the inside the egg cell, the outermost loosely bounded cytoplasm are also having some kind of a protein, the motor protein, like cytokine skeleton, like microtubules made up of a tubulin protein, like microfilaments made up of actin proteins. Whereas the inner core part is something very tightly bound cell cytoplasm is there. The outer cortex I have already discussed have loosely bounded cytoplasm, helping them for mixing. That is, the actin protein helps in the formation of a microfilament, microtubules made up of a tubulin protein cytoskeleton that also maintain the shape of the cell. However, the inner cortex are tightly bound with dense cytoplasmic content. The cytoskeleton arrangement occurs through the help of the motor protein, and the cytoskeleton in the cortex is modified easily than the inner cortex. Okay, how the cellular movement or the cytoplasmic movement takes place? Cytoplasmic, always remember, the cytoplasmic movement takes place using the help of certain kind of a motor proteins, like actin proteins, like microtubules, cytoskeleton, that is important for the proper development of an embryo. Now, there was some experimental procedure was done by scientists, early embryologists like Holliston and Allison. That is the formation of a belly piece. Means in the developmental term, if an abnormal piece of mass formed, that is known as a belly piece. Okay. Till then, the scientists did not know what leads to the mixing, what leads to the formation of the embryo. Whether it's the component, morphogenetic component in the cytoplasm, or what makes it, or the cytoplasm itself, what is the important factors for making the this proper embryo. Then they have done two sets of experiments. In one set of experiment, they have taken again two zygote. One was experimental zygote, and of course another was a control zygote. In the experimental zygote, UV radiation was exposed to the vegetal hemisphere. Okay, means to the dorsal side. In order, why? Because out here, see in the vegetal hemisphere, what is there? Huge numbers of wide range of morphogenetic gradients are there, because dot concentration is less. Okay, so UV radiation was exposed in the vegetal hemisphere in order to destroy the vegetal hemisphere. Means ultimately to destroy all the morphogenetic components like cytoskeleton, like everything, all this, like microtubules, microfilaments. Okay, then they have seen that when they have exposed UV radiation in the vegetal hemisphere, in the control, no such UV radiation was exposed. At that time, they have seen that there was a formation of an abnormal piece of mass, that is the belly piece in the experimental zygote. Then that of the control. Control was quite normal. Means normal embryogenesis has taken place. Now out there, they were not clear what led to the formation of this normal embryogenesis. It's the cytoplasmic mixing or it's the like the presence of the morphogenetic gradient. They were not clear. So in the next set of experiment, what they did, they have UV radiated, exposed both the experimental cell also, control cell also. Means they have taken another cells. One is experimental cell, another one is a control cell. In the vegetal hemisphere, they have UV exposed both the cell, experimental also, control also, in the vegetal hemisphere. After that, after UV exposing, they have fixed the one of the cell. They have fixed one of the cell. Sorry, they have fixed both the cell in the gelatin. They have fixed both the cell in the gelatin. When they have fixed both the cells in the gelatin, the cortex part of the cytoplasm, which was already very fixed, had become solidified. However, there was few of the cell cytoplasm outside which has not yet attained the solidification or the calcification. Now, what they did, they have turned one of the experimental was remaining one of the experimental zygote. They have kept it in the same position, it's 90-degree position in the gelatin mix. Okay, and another control they have tilted to the 45-degree angle. They have tilted to the 45 degrees angle. And they have noticed that the development of a normal embryogenesis takes place when they have in which they have tilted to the 45 degrees. Means when they have tilted at that time also what happened? The inner cytoplasm has moved. The inner cytoplasm has moved from the animal hemisphere to the vegetal hemisphere, little bit this side and little bit somewhat in this side and somewhat from the vegetal hemisphere to the animal hemisphere, the other side. Okay, from there they have confirmed that the like the proper embryogenesis takes place not only because of the morphogenetic gradient, not only because of the morphogenetic component, it's because of the mixing of the cytoplasm. Because see, they have already UV exposed, they have already given the UV radiation to both the control and the normal cell and like experimental cell. If it would have been the morphogenetic component which would be responsible for formation of this proper embryogenesis, then knows both it should have developed both the experimental also, control also as a balance. But what we have found that it was the tilting. Tilting has led to the formation of a proper normal embryogenesis, not the formation of a balance. So from this experiment, they have confirmed that the cytoplasmic mixing is quite important for the formation of a balance. Okay, that's all for today. Thank you.